Chemicals that combine to enzymes and eliminate or drastically reduce their catalytic activity are called enzyme inhibitors. Enzyme inhibitors are classified on the basis of whether the inhibition is reversible or irreversible, competitive or non-competitive. Reversibility deals with whether the inhibitor will eventually dissociate from the enzyme, releasing it in the active form. Competition refers to whether the inhibitor is a structural analog, a lookalike, or a natural substance. If so, the inhibitor and substrate will compete for the enzyme's active site. We're going to look at two types of inhibitors. Irreversible inhibitors, those are ones that bind tightly to the enzyme and thereby prevent the formation of the enzyme-substrate complex. And reversible competitive inhibitors. These often structurally resemble the substrate and bind at the normal active site. Irreversible enzyme inhibitors, such as arsenic, usually bind very tightly, sometimes even covalently, to the enzyme. This generally involves binding of the inhibitor to one of the R groups of an amino acid in the active site. Inhibitor binding may block the active site binding groups so that the enzyme-substrate complex cannot form. Alternatively, an inhibitor may interfere with the catalytic groups of the active site, thereby effectively eliminating catalysis. Irreversible inhibitors, which include snake venoms and nerve gases, generally inhibit many different enzymes. Reversible competitive enzyme inhibitors are often referred to as structural analogs. That is, they are molecules that resemble the structure and charge distribution of a natural substrate for a particular enzyme. Because of this resemblance, the inhibitor can occupy the enzyme active site. However, no reaction can occur and enzyme activity is inhibited. This inhibition is competitive because the inhibitor and the substrate compete for binding to the enzyme active site. Thus, the degree of inhibition depends on the relative concentrations. If the inhibitor is at excess or binds more strongly to the active site, it will occupy the active site more frequently, and enzyme activity will be greatly decreased. On the other hand, if the natural substrate is present in excess, it will more frequently occupy the active site and there will be little inhibition. This figure represents reversible competitive inhibition. So you see the topography of the binding site in the enzyme, and it actually matches both the normal substrate and the competitive inhibitor, but you can tell that they're two different molecules. They're very different, the red molecule and the blue molecule, even though part of each of those molecules does fit the active site. Well, if the normal substrate binds to the enzyme, then you get your reaction proceeding. But if the competitive inhibitor binds there, and why would it bind? Well, if it's in higher concentration or if the enzyme bonding pocket has a higher affinity for the competitor, what it's going to do is it's going to block that active site.